Yeah, thank you very much for attending this workshop of Molecular Informatics team of RIKEN AIP. And actually, this meeting is also broadcasted by Zoom, and I saw more than 70 people signed up, so I'm very happy about that. And my name is Koji Tsuda, and I'm a team leader of this molecular informatics team. And now I'd like to give you some introductory talk about our team, and actually technical part will be presented in next talks. And first of all, I have to start from what is RIKEN AIP. Actually, RIKEN founded Center for Advanced Intelligence project in 2016. And it has like more than 100 employee researchers and so on and so forth. And actually, it's quite international center where you have like half of um, researchers are like non-Japanese. And now, initially it was planned to last 10 years. So this should be the last year, but it now extended to 2032, so we can do it a bit more. And actually, there's a lot of teams. One is so-called generic technology, which is for theory of AI, and the goal-oriented technology teams for kind of applications of AI, and AI in society teams for like ethics kind of stuff. And actually, first group of teams are developing new AI technology like theory of deep learning and machine learning methods itself. And actually, the second one is also, it has activities of accelerating scientific research, and our team is one of them. And it spans from like medical science and material science, and maybe astronomy, and even music, and so on and so forth. Yeah. So actually, there are some teams that are engaged in natural disaster and elderly healthcare and education and so on and so forth. So it's kind of for social science, and also you have study of AI in society where you kind of help Japanese government to have ethical guidelines of AI, etc. Yeah. And actually, this is financial year of 2025, and this is the last year of the first term of the IP. So we need to kind of wrap up our activities in these 10 years, and actually every team has to do this kind of workshop to wrap up the activities in these 10 years. So and this yellow one is now here. Okay, then I go to the main part. Yeah, so our team is called Molecular Informatics team, and I want to talk about some activities we are doing now, and also I want to discuss a bit about future of this team, and actually, mostly, I mean, we have been working on those two things. So one is analysis of protein dynamics with rigidity theory, which is mainly done by Adnan, and another is using AI to design molecules and matter, mainly by Sumita-san and me and others. And actually, about the future, I want to first discuss about the impact of large language models to AI for science, because LLMs are now kind of so powerful, and then it's not negligible, and also I bit talk about algorithms for self-driving apps. Okay. Okay. So I briefly talk about the first part. So this is Allostery analysis by rigidity algorithm. And actually, when you have protein, the structure of proteins now can be solved by this AlphaFold, which won Nobel Prize very recently. So this is very famous. But the most important thing about protein is dynamics. So, for example, if a ligand, small molecule binds to part of protein, then it changes. The structure changes. And actually, this change happens kind of slowly. So for example, when one molecule binds at one end, then this is kind of transmitted to the active site. But it takes quite a long time, like 1 millisecond or something like this. So it's very hard to simulate by molecular dynamics algorithms because it's too long to because the phenomenon happens in a longer time scale. And what we are doing is to represent protein structure as a framework. So in this kind of basically graph. And it's a graph where each node is atom, and the edges are either covalent bonds or hydrogen bonds. And actually, by using so-called rigidity theory, you can identify which part is rigid in this kind of framework very fast. And then once you can identify the rigid part of the protein, this reduces a lot of degree of freedom. So think about it like if you have 100 atoms, then there are 300 degree of freedom. But when you identify everything is rigid, then you end up with 6 degree of freedom, right? So this is very, very huge reduction. And by doing so, you can do this ultra-fast dynamic analysis. And actually, it has been found, and actually, Adnan was collaborating with Professor Prosa, today's guest, for a long time, and they proved that this approach is really accurate, really accurately predict the behavior of protein using NMR techniques. And for example, if you see some protein called A2A, which is a membrane protein, and actually it binds to 2 different so-called G proteins. And if you see A2A binding to different proteins, then the rigidity pattern changes. And it means that the signaling of this protein changes a lot, and this has a lot of significance when you think about like drug discovery and so on, because essentially you have to find a small molecule binding to this GPCR type of proteins. Okay, so this is one thing, and maybe you can see more in the next talk. And like the second thing we have been doing is this automatic materials design. So what we have been doing is to automate materials design procedures. Like usually you have human scientist here, and then he or she comes up some kind of materials, and then experiments are done, and then you get data back. And then after that, you rethink and come up with another material and then do another experiment and so on. So by doing this loop, you can develop material design. And what we want to do is to replace human with machine learning to do this automatic materials design. And of course, if you can replace experiments with simulation, like quantum chemistry simulation, then of course you should use it because it's much faster and you don't need to do actual experiments. So and we have been using DFT. DFT is not a discrete Fourier transform. That's DFT. Sorry, density functional theory. Yeah. So it's type of quantum chemistry simulations. And actually, yeah, so in this 9 years or so, we have been developing a handful of tools for materials design. Maybe yeah, like KMTS and Fizzball and so on and so forth. And I'd like to introduce mainly 3 studies. All of them are collaboration with experimental scientists. So not only we create materials by machine learning, but they are always validated by experiments. So first of all, what we did is to develop wavelength selective thermal radiator. So actually, it looks like this. Um, and what it does is that given heat from below, so maybe you have some fire or something, and then it emits light of certain wavelengths, and we want to control the emission spectrum so that it is concentrated into some wavelengths. And this type of material can be used for solar absorber or heater, like let's say so like heater for like heating up or drying and something like this. And you can use it for sky radiator where you can emit the light that that the air cannot absorb. Then what you can do is to throw away heat to the outer space. Yeah. And what we did is to come up with this kind of layered material. So in this case, we had 18 layers, and the idea is to use germanium or silicon or silica in each of the layer, and actually you can also control total thickness of this layer, and um, and if you think about it, so the number of candidate structures is 3 to the power of 18 times 21, so it's about 8 billion tons. So actually, yeah, you may do like experiments for all of these 8 billion candidates, but it's impossible. So we used technique so-called Bayesian optimization that so that machine learning suggests you reasonable layer configurations. Okay. And as a result, we came up with optimal. So given the target spectrum wavelengths, we came up with this kind of structure. And actually, it's not kind of periodic. I mean, it's quite asymmetric and complex. And we could do that within, say, 1,000 simulations. Sorry, we are using certain simulations to do that. And actually, this is the computational spectrum of the created material, and you can see that it's almost perfect because it's peaks are really sharp. And if you actually made it, then it looks like this. So it's less sharp because it's actually measurement, but it's still very, very sharp, right? So this was very influential work, and I think it's published in 2019, and now you have this kind of work everywhere, maybe more than 100 papers doing very, very similar thing using machine learning. Okay. And by the way, this is a collaboration with Professor Shiomi, who is talking this afternoon. And next is molecular generation. So basically, what we're doing is to combine, say, generative AI with simulators like quantum chemical simulators, and we can design some molecule which have desired properties, and properties are computed by such such simulators. And actually, what we did is to develop fluorescent molecules, like shining molecules. And to do that, you need to, you need to simulate fluorescent molecules. And to do that, you need to follow the path of this absorption, relaxation, and fluorescence using density functional theory calculations. And so it's not so easy, but it's done in large scale. And finally, we came up with these 5 promising molecules. And this figure is just about, I mean, yeah, so it's, yeah, I mean, it's not sure, but basically the thing is that we generated more than 3,000 molecules on the computer using more than 1,000 cores, CPU cores for 5 days. So actually to do that, this is very important to do simulations on large scale, not just once. So it has to be automated, and this is also very nice, important aspects of this. And we validated, we synthesized and validated this molecule called PC, and it's found to be actually a fluorescent. Yeah. So this is the first fluorescent molecule found by AI. And this type of molecule generator is also used in drug discovery purposes and so on. So yeah. So this was also influential work. And thirdly, in collaboration with Professor Umetsu, who is also speaker today, we are doing protein engineering using machine learning. So in very, very simplified way, so the conventional protein engineering is written like this. So you have some existing protein, and then you give proteins random mutations. So you just randomly change some specific part of the protein sequence, and then you do measurement, and you select good proteins. And then after that, you do this again. So basically, you pick up one good protein, and then do random mutations again, and then give it measurements. But actually, because it's random, so it's just random mutation, so it may not be so efficient. So what we did is to do this once. So we do conventional thing once, and then we get some initial data, and then we run a Bayesian model, like Gaussian process model, and we design new proteins based on this model via Bayesian optimization. And then we actually created using E. coli, and I mean in MedSense's lab, and then we did measurements, and we had good proteins. And yeah, and actually what we did back in 2018, so actually there are a lot of studies recently about protein design, but we did it really early. I mean, in 2018, we designed fluorescent proteins by kind of modifying GFP, green fluorescent protein, and so that it becomes more yellowish. And then we succeeded to make it actually yellow, like this. And based on these results, some startup company was founded, and now they are using this technology to their business. Okay, and this was like some summary of what we did in these years. Yeah, it's only part of that, but maybe you can hear more in next talks. And I'd like to talk a bit about future, and especially impact of LLMs to AI for science. And actually, before LLM, what we did is, what we have been doing is something like this. So basically, we have several data sets, and we essentially select one data set or two, and then we design some kind of deep learning model, and then solve certain task, say molecular generation task, or property prediction task, or something like that. But actually, there are many data sets of different kinds. And for example, when you think about biology, you have gene, and the gene is transcribed to RNA, messenger RNA. Messenger RNA is translated to proteins, and the proteins have functions, and also, maybe in MR spectrum and so on, or, evolutionary information and so on and so forth. So when you have some scientific object, you have really multiple data set, and unless you integrate them all, you cannot really solve the problem, actually. And we knew that, but actually there's no way to do that. But actually LLM may give us some chance to do that, because actually when you describe each data set in natural language form, and then if you fine tune some pre-trained LLM like those by OpenAI or something like this, and then maybe you can integrate all the data set into one model, and solve all kinds of tasks at hand by kind of commanding LLM to do that. So that's very attractive opportunity for us. And actually, what's happening right now, I mean, given LLM, is the diminishing value of models. So basically, we have been developing deep learning models for this and that and so on. But if LLM can solve it all, then why do that? Actually, that's the question. And another thing is skyrocketing value of LLM ready data sets. So basically, now you can make use of all kinds of data at once. But actually, you have to make it LLM ready, and it means that yes. And then what happened is now there are lots of benchmark papers, so-called, and this ChemBench, for example, which is published in Nature Chemistry maybe last week, is all about like representing all kinds of chemistry problem in natural language and make question and answer data set. So that's all about it. So basically, there's no machine learning, or there's no new models or something, but this benchmark is all about Q&As and natural language representation of data. And actually, we are doing kind of similar thing for other issues. Yeah. And another question is how you given some data set, how do you give it to LLM? So it's not so easy to do, and you may need new representation to do that. And this is another thing. GPT is what we are doing right now to this purpose. Maybe too limited data set, but yeah. And actually, what ChemBench, I mean, it's not our work, but what ChemBench is doing is they created natural language Q&As from various chemistry data. So in a sense, you can do this. Part of it, you can do this automatically. Like maybe it's hard to see, but this is what is the number of signals in the 1H-NMR spectrum of this molecule on the right. The answer, you prepare the answer, and then and then you can make this kind of data set, and then LLM you ask this question, and LLM can answer, and it may be right or wrong, and then you can kind of give LLM some score and say, okay, this LLM was like the best, and this was second, and so on. So you can use it for LLM evaluation, but more importantly, it can be used for training, right? So maybe you don't need Q&A data set for training, but what's very important about training is that you have to evaluate how LLM understand it, right? So that's so therefore Q&A are definitely needed. And recently we made a benchmark data set for nanobodies, like nanobodies, very specific antibody found in camel, where you use only one protein to make an antibody. But this is what we did very recently, and actually, yeah. Another thing what we did is about peptides, and what we did is to find non-canonical amino acids from Chembl database. So in Chembl database, you have chemical formula of peptides, including non-canonical amino acids. So we found it, and then we defined tokens for non-canonical amino acids, so that you can train LLM using this new representation. So this is one kind of thing you can do. But actually, yeah, actually there are lots of databases out there. So, but they are not really LLM ready. So there are a lot of things to do for us. So and actually the question is if we can really really do this type of data integration and reasoning across data sets. So that's the question we have, and we still don't know really. Okay, and another thing we are planning to do is to develop algorithms for self-driving labs, and actually, yeah, we have collaboration agreement with NIMS. NIMS is National Institute of Material Science, and they have this kind of machines to do automatic experiments. So, and we are developing suite of machine learning algorithms called NIMO, and that contains our own algorithms, and we are trying to make more algorithms and integrate with it. Okay. Okay, so conclusion. So I said several things, but I will continue. We will continue and expand existing success directions, like protein dynamic studies and AI-based design materials. But actually, new directions. As new directions, maybe we need to adapt more to LLMs, and self-driving labs are also very important aspect. Thank you very much.
Thank you, Suba-sensei, for your talk. In my lab, we're interested in a class of molecules that are fluorescent, but also solvatochromic.
Oh, really?
So,
we are now really working on this what was that, Sumita-san? Solvatochromism, right? Yeah, so we can talk later. Yeah. I mean, that's yeah, it happened. Yeah, it happened to be the topic we are working on right now. So, one question from Zoom. There are proposals like AI for science utilizing LLMs and evolutionary training. AI scientists at Sakana AI and Fireball at Google, etc. Will these become mainstream approach for AI for science in the future? Maybe. Maybe. Maybe, but actually scientific studies. So the question is, I'm not so sure about it actually. But obviously, the good news is that now we can combine all kinds of data without difficulty. Once you translate into English, everything is into English, then then you can just combine all data to big file and then give it to LLM. So that's really great thing, because we tried before. We tried to do this kind of thing with other techniques, but never successful. Yeah. So yeah, so we want to kind of evaluate it using our own data. And another question is, what areas should we focus on further research in order to implement a fully automated materials discovery pipeline system using AI agents, including data collection of both data and specialized knowledge, experimental design planning for predictive model construction, experimental instruction, data analysis, yeah, and paper writing. Well, okay. Yeah, I mean, that's a very interesting question, but actually the research about self-driving cars, now, kind of directionless. So basically, there are lots of things to do, and actually, making hardware is really hard. So so actually, it's not only one thing, but there are many things to do, and right now it's very hard to kind of focus on really one thing to make breakthrough. But certainly, self-driving graphs are very important topic, and certainly there are a lot of labs in the world doing this right now. So maybe in this year or next year, you can see some kind of directions. While university, including the University of Kyoto, have announced plans to begin constructing a curable materials database, could you explain the necessity, feasibility, availability of such a project? So this kind of project, like storing all possible data, I mean, so this was, I mean, I hated it because actually so far there's no point in just having data with like all kinds of data at once. But because LLMs emerged, this might make sense now. So this is a really big change, and finally, this kind of project might make sense.
Thank you. So I'm Adnan Sljoka from RIKEN AIP, and I'm going to be discussing how we can probe protein allostery and functional dynamics using techniques from rigidity theory, NMR, experimental data, and time permitting a little bit, I'll mention reinforcement learning. So what we're really after is we're after current limitations in experimental structural biology, particularly X-ray crystallography, cryo-electron microscopy, or even these AlphaFold predictions give you kind of snapshot representations of proteins, but they don't really reveal the dynamics that is really critical to understand how proteins function. And what I want to further argue is that it's not only the dynamics it's important, it's the regulation of dynamics when the protein interacts with, say, different ligands, drugs, cations, or different proteins, in particular through these kind of long-range allosteric mechanisms and some methods and algorithms that I've developed over a number of years that is really turning out to be extremely useful. So first, I'll, for those of you who are not in the area of protein research, I'll just introduce some basics of why we care about flexibility dynamics, and really the future of AI applications in this work is to look at dynamics and allostery. And then I'll introduce briefly what is rigidity theory, just to give you some kind of a flavor, and how we can extract algorithms such as the one I developed, rigidity transmission allostery, and various different algorithms for probing these allosteric mechanisms, which has been known as the second secret of life for like 67 years. Meanwhile, it has really huge potential in, say, drug discovery. It's completely very not used enough in drug discovery. And then I'll mention some studies like on G protein coupled receptors, just because of to give some more computational and context to the work in collaboration with Scott Prosser. There's other things I could say, time permitting, like importance of water networks in allostery and allostering drug discovery, and some new efforts we're trying to do now to extend some of these AlphaFold predictions for refinements and for disordered proteins. So as I said, I'm just going to discuss mainly the work of prediction of allostery and applications on G protein coupled receptors, but there's many other things that I'm interested in combining various computational experimental techniques. So if people want to discuss after more about structure validation or disordered proteins, or even allosteric drug discovery, then happy to do that. And of course, the long-term goal of this is to kind of have automated discovery and design of proteins and allosteric drug-like molecules. So proteins are composed of a sequence of amino acids, and then they form in some kind of a shape or structure. So once we have the structure, we'd like to be able to understand its function, and of course, in applications like drug discovery. And of course, as all of you know, these tremendous advancements in AI and deep learning now enable us to get an enormous amount of structural information, something like 200 million predictions. Of course, there's still a question of how good these structures are, but the bigger question is that we would like to be able to have even algorithms that can handle, or methods, this enormous amount of computational structural data. However, to really understand how protein functions, as I said, we need to understand flexibility and dynamics. And moreover, what we're really after, what I'm really interested in, is how this dynamics is regulated when a protein interacts with a drug or another protein, and how sometimes this, in many cases actually, can propagate through different regions that are functionally important and modify the function of the protein. So as I said, the main limitations of structural biology, experimental techniques, and even these AlphaFold models, which were trained on these data sets, is that they give you these kind of these pictorial representations of proteins in low energy states. However, biologically relevant conformations that, as I'll show later, and maybe in Scott's talk you'll see, are not necessarily the most stable. So what you're really after is some kind of higher energy states. And to capture this, you need to really be able to understand flexibility and dynamics. And the big picture, the big message is that we need really fast algorithms to handle this enormous amount of data. So just to give you some motivation of some of the questions that we're after, I'm not going to talk about all of these, and these are just some number of studies that were published. So you can see here a receptor depicted on the left. These are some of the major drug targets. It has a lot of dynamics, and then when it interacts with its partner, some of this dynamics is lost. And there are other further questions. How are these kind of allostery and is being modified when you have these kind of interactions? These kind of receptors can interact with cholesterol, calcium, and magnesium. So how do all of these perturbers, how do they affect dynamics? How do they affect flexibility? How do they potentially affect allostery? And then more advanced topics, we also know there are water networks, not only on the surface, but can be found buried inside the protein, and this can modify the interactions and the allostery. The other thing is that, for all of this, we need some kind of input, some data. So the structures that we're working, whether they're experimental structures or these AI-based structures, we need to know that they're valid. A number of years ago, I developed some methods using rigidity theory and experimental NMR to validate the quality of NMR structures, because it's really been difficult to see how good NMR structures are, unlike crystals or cryostructures. And in fact, using these kind of techniques, we can actually extend some of these methods and combine with reinforcement learning to say, to improve or refine AlphaFold structures. And you can see there's this kind of image that AlphaFold always gives you the right structure, but there's a number of examples, particularly on these kind of smaller proteins or disordered proteins, where it can get grossly wrong predictions. So using these kind of techniques, we can further refine even AlphaFold predictions. And in RIKEN, there's been a long interest in NMR structure determination, and these kind of tools are becoming really powerful and useful to further validate or improve NMR structures, but also these AlphaFold models. So again, just to give a little more background, why is flexibility and dynamics really difficult to study? Well, one of the reasons is that the motions occur in so many different timescales. And in particular, these longer timescale motions, milliseconds, seconds, is where often many biologically relevant processes like allosteric regulation, GPCR activation, enzyme catalysis occurs. However, if you want to just classically simulate the protein motions using molecular dynamics, it's very difficult to probe these longer timescale motions. So that's why we're after kind of alternate techniques. And it turns out rigidity theory, as previously my colleagues and myself have shown in a number of papers, can probe these longer timescale motions. So this is where it becomes also really powerful. Plus, the additional reason is that these methods are extremely fast. In fact, this was part of originally of an older project where we were after very fast algorithms for a number of different applications, and rigidity theory we try to, I developed many algorithms in rigidity theory that make them even faster. So now, of course, there was no AlphaFold predictions. This was like 6, 7 years ago, but now it's really become relevant, this kind of work. And in fact, there's some kind of surprising connections in mathematical, because there's a lot of, the rigidity theory is really deep in mathematics. Things like maximum flow has some nice, very connections to prediction of rigidity and how we can even further speed up these methods. So rigidity theory is quite rich area of mathematics, and just to give you kind of a glimpse of what we're even after here. So rigidity theory is concerned with rigidity flexibility of structures that are defined by some kind of geometric constraints. These could be length, angles, directions, on some set of geometries. And really, it's a really rich history in mathematics. Unfortunately, it's not accessible to most people in applied science because of this deep mathematical background of it. However, over the last 30 and 40 years, the number of theoretical and algorithmic results that I've developed allow us now to apply these in various different areas of applied science. Today, of course, I'm talking about proteins, but I could equally be talking about linkages in robotics or even in material science. So a lot of the goal of my research is to kind of take this deep mathematics and the background I have and to connect it to various different applications. And in many ways, rigidity has already been disruptive, for example, in material science and design of these Gorilla Glass, sold in over 8 billion devices, where you design these kind of more stable devices for your mobile phones. So what we're after is you have some kind of framework where you have some kind of constraints between them. This is very simple. Unfortunately, calculating rigidity is extremely difficult. However, there is one aspect of rigidity, it's called combinatorial, where you assume that these positions of these points behaves in some nice geometry. Once you do this, it turns out the rigidity is only a property of the underlying topology graph. It's completely remarkable result. And what this allows you to do is it allows you to develop fast algorithms. So for these kind of simple structures, this actually extends to robotics-based structures, molecular structures. We can take these kind of PDB structures, turn them into these molecular graphs, and apply these kind of combinatorial graph-based counts. So what it just says is that you need to apply certain combinatorial counts. This was conjectured a while ago by Tay and Whiteley. I was fortunate enough to do a PhD with Whiteley, who was really pioneer in building up rigidity theory. And we can develop really fast algorithms now to capture the flexibility and rigidity of these kind of structures. And again, you can imagine, just besides proteins, that this can be applied in, say, material science. So there's many things you can do. You can screen, for example, a lot of drugs, see how the drugs are affecting rigidity and stability. You start with the protein structure. You build up these kind of various constraints. You can correlate this to experimental data like NMR. And I've shown a number of papers that we can analyze a large number of structures, something like hundreds of thousands of models and structures like antibodies, looking at these binding CDRH3 regions, or analysis of all proteins in the PDB, as we did here for NMR structures. So because these methods are extremely fast, and the reason they're fast is because of that underlying combinatorial theoretical characterizations of rigidity. Moreover, should play a movie. Moreover, you can actually start to simulate some of these motions. Once you know where these rigid, flexible regions are, you can reduce the conformational space, and you get something like 100,000 times faster than MD simulations. In fact, we can even start analyzing disordered proteins that have an enormous amounts of conformational degrees of freedom. And some of the efforts now is to use experimental data like SAX and NMR, combine it with rigidity, and kind of modify and fix some of these AlphaFold predictions on disordered regions of the protein. And just as a reminder to those working in material science, as I said, these algorithms can be adapted for materials. So let me now just introduce this notion of allostery. So this has been called a second secret of life. It's been around, I guess, from 1965, probably even prior to that, in something called Bohr effect. And what it does, it refers to regulation of a protein function at a distance. So if you imagine your protein is here depicted in this blue cartoon here, and you have some kind of event, maybe ligand binding, maybe mutation, and this can actually locally change your dynamics, but it can propagate and change the shape and change the dynamics at these distant sites. And how this occurs has been kind of very poorly understood. But it's really critical for in control of metabolic signaling pathways, diseases. Many cancers are turning off through these kind of faulty mutations that can be away from the active site. So there's a huge interest in this and understanding, of course, and this also has a huge potential allosteric drug discovery if people are interested and are happy to talk about that. So I've developed some new aspects of new ways to think about this, first of all. So before I even started talking about proteins. So imagine you have this kind of mechanism and you have this slight change in shape at one site. This can propagate, and the shape can propagate to these different regions. Or in these kind of more complex networks, you can see this kind of similar effect. It turns out, I've developed some theory that if you rigidify one of these sites, state A, and you change the local conformational degrees of freedom, this can propagate and change degrees of freedom at these distant sites. And it turns out actually that mathematically that's a statement that this degree of freedom propagation is equivalent to change in shape at these different distant sites. So just for, those are not in the area of protein, just to give you a visualization of what we're even talking about. So you have so many possible shapes being sampled. This just has 3 degrees of freedom. You restrict just a couple of them. And now you're forcing this one on the right to move in a very specific way. So it's directly kind of inherently capturing this idea of allosteric transmission and conformational selection. And there's many algorithms that I've developed for capturing these kind of ideas, and I don't have time to go through them. So if you, for example, scan a protein across the perturbing each residue, and then you do this, you get these kind of predictions. So you have these local changes here. Oh, you can see. You have local changes in the red, but then sometimes you have these regions that propagate quite far away. And this actually correlates to some NMR experimental measurements like chemical shift perturbation. And we show this in many structures. So meanwhile, history of Allostery has been, as I said, a long time around. There was this review. So using these kind of rigidity-based predictions, we can begin to understand allosteric mechanism. And I was fortunate to be at the conference and give a talk where Jean-Pierre Chenjou, who introduced this MWC model, was there. And we're now having some discussions how to extend these things further. So there's many challenges also in allostery. And this is where people in drug discovery often fail and they don't understand why. So let me just give some one appreciation of why this is challenging. So you can see here, there's a kind of opening motion leading to an opening motion, or closing. And if we just break some of those hydrogen bonds, now we can get an effect where it's an opening motion leading to a closing motion. There's a different effect that occurs here that you saw before. And this is just by changing some of these hydrogen bonds, or breaking some of these bonds, making them flexible and making some other ones rigid. So we can really kind of fine-tune this effect. And that's where allostery is extremely powerful, in this fine-tuning the effect of actual drugs. So let me give some examples of some of these ideas on actual protein structures. And I'll focus on GPCRs just to give maybe context to later talk by Scott Prosser. So GPCRs are the largest family of cell surface proteins. There's over 800 GPCRs in humans. There's different classes of GPCRs. About 40% of all drugs actually bind to approved drugs. And yet less than 20% have been drugged. And one of the reasons is that we really still have a poor understanding of their activation and these allosteric mechanisms. They're really naturally allosteric. So the way they're activated, you have this inactive form of a GPCR. You have an agonist that binds, and that often leads to some conformational change in shape, and then it can bind to its main partner G protein. And there's many kinds of other G proteins. They can be promiscuous. They can bind to other things like arrestants. But even this kind of visualization that I just showed you is kind of grossly wrong. Yet we have many structures in the inactive, in the active states. It really doesn't answer things like how they can couple to multiple G proteins, something called promiscuity. And in some cases, you can have a ligand that binds to the same site, and another ligand binds to the same site, yet it has a completely different effect. So how this occurs is poorly understood. So in collaboration with experimentalists and particularly with Scott Prosser, he's got these beautiful NMR experiments, and maybe he can explain more detail. But what they do is they label certain residues, and they attach this kind of 19-fluorine tag, and they can manage to see how the conformational change as a protein goes from inactive and active states in the presence of different drugs, different G proteins. And sometimes these transitions occur, as I said, on tens or hundreds of milliseconds. So performing MD simulations would be extremely difficult to capture this. So here are just some model studies that we have done. For example, we were asking so MD simulations show us, so these GPCRs can interact with cholesterol, they can interact with calcium, magnesium, sodium. So here's some simulations showing that calcium, magnesium can bind at these locations. And I've shown that these actual parts here are kind of allosteric, which you expect this information down to transmit. But furthermore, when you restrict some of these conformational degrees of freedom with these calcium, magnesium bridges, you increase this allosteric effect. And under maybe very high, not physiological conditions, experimental data has actually shown that you can shift these populations to more active-like conformations. So we're basically interested what other perturbers of this structure can have these kind of allosteric effects, and what are the role in the dynamics. The one question is that, of course, this GPCR has to interact with its G protein or beta-arrestin or other, but the G protein itself has different subunits. And a lot of studies focus on this G alpha because it's the main, it interacts directly with the GPCR. However, it's a little bit unknown what the beta gamma may be doing. So what we wanted to know is, does the beta gamma influence the activity and potentially dynamics in allostery? So NMR data, and again, I'm not going into detail, has shown that when you have beta gamma versus when you don't, you get more access to these active-like conformations. And similarly, functional studies have shown that if only you have G alpha versus G beta gamma, you get much better rate of something called GTP hydrolysis. So really suggesting that this subunit on the right is kind of some kind of allosteric stabilizer, and that may result in allostery. And that's exactly what these rigidity-based predictions have shown, that you get this allostery going all the way from the yellow ligand down to the green GDP pocket. Moreover, you can see that the right subunit, the beta-gamma subunit, is also involved. And in fact, if you remove the beta-gamma subunit, you get weakening of this allostery signaling. So just to give you some appreciation now of how we think this may work. So you have this receptor that's quite flexible, quite dynamic. Even without any G protein or ligand, it can still become activated. However, when it interacts with the G protein, you get this reduction in rigidity and stability, and you get this emergence. You saw this white parts. These are the allosteric pathways. And then when the full agonist binds and engages, then you get emergence of these much, much stronger allosteric pathways. So this is still much, we have deep understanding of this, but this is still simplistic. These allosteric pathways themselves are quite dynamic and are changing. And then this causes other kind of conformational changes that are needed. Not all perturbations of the structure are necessarily allosteric. In fact, here's a study where we showed that when you look at cholesterol in many crystal structures on this GPCR called A2 adenosine receptor, so this is the one I'm showing in all of these examples, can be bound here. But these experimental data, you can see here as you increase concentration of cholesterol, you really don't have this change, significant change in GTP hydrolysis. Also, similarly, this conformational 19F fluorine NMR data doesn't show any major changes. And remarkably also, if you have cholesterol, if you don't, as a perturber, it doesn't change the strength of this allosteric coupling, as shown by rigidity. As I've said, these GPCRs can also be promiscuous. They can bind to different partners. In this case, this A2 adenosine can bind to GS and GO. So we wanted to understand how it can bind, even in the presence of the same drug, same agonist. NMR data, again, I will just kind of go over the basics, has shown that there are a couple of these active-like states, and they can be differently populated in the presence of GS and GO. And there's also some key differences in dynamics. And we verify this with MD simulation and Monte Carlo simulation. So, but again, kind of hinting that there may be some differences in the way these signals are being propagated across the structure. And that's exactly what we found. So in the presence of GS, this allosteric pathway is different than you can see in the presence of GO. And there are some key known prior kind of so-called allosteric switches that are being engaged more in the presence of GS and GO. And we've shown similar kind of results for some other receptors, and we're trying to extend this to other kind of GPCR. So really, allostery is critical for this kind of promiscuity, for understanding how they bind to different proteins, to bind to GS or GO. I could talk about allostery in other systems, but I'm running out of time, so I'm just going to maybe quickly mention some key things we're thinking about. So this is an enzyme. It's a homodimer enzyme, and it breaks one of strongest bonds, this carbon-fluorine bond. And what it does is that when the substrate actually binds only to one monomer, and when the substrate binds to one monomer, it actually increases the dynamics of the second monomer. But if you look at these structures across from the apo structure, the substrate bound structure, the product bound structure, you really don't see much difference among them. So it's really difficult to depict what are these different dynamics and allostery. So most of the difference are because of some of these side chains are being also changed, and therefore the underlying hydrogen bond network is being modified. But this is difficult to see if you just superimpose these structures. However, rigidity again finds there's a clear allosteric pathway, and similarly NMR data verifies that there's changes when you substrate binds, there's these increases in this NMR, so-called chemical shift perturbation. Sometimes when you increase concentration of substrate, the substrate itself can act as an inhibition by binding to a kind of a second pocket along this allosteric pathway. But one of the things we're really thinking about now, and not just for enzymes, but even GPCR in general, is that often you'll get waters that are found, as I said at the beginning, at the kind of a core of the protein structure. Waters, of course, can form hydrogen bonds. So you would think it would change rigidity and flexibility and potentially these allosteric networks. For this enzyme in particular, crystallography data shows that when the substrate binds, there's like a loss of 30, 40 water molecules in the second empty monomer, suggesting reorientation of these waters. And as you can see here, as I go here, showing you these waters, you can see that the waters are actually changing themselves. Of course, now gets into that complexity that I was talking about. Allostery, this may be modifying the allosteric networks. And indeed, when you rerun this analysis plus minus waters, you can see there's a modification in the strengths of this allosteric coupling. And maybe Scott may talk about some experimental data for supporting this. And you can now do all sorts of things. You can look at coupling between different pairs of residues. In this study, we correlate this with two-dimensional NMR. In another study with collaboration with Polarino at OIST, completely unrelated application, but it's very relevant to, say, enzyme evolution and enzyme engineering. It's an interesting enzyme that when you insert some of these remote loops, one of these loops is allosterically coupled to the active site, and this remote loop insertion leads to acquisition of a completely new activity, in this case, antifungal activity. Meanwhile, it still maintains its original enzyme activity. So allostery really can be applied and has many fascinating applications. We're also working on understanding, for example, spike protein in coronavirus. This is just accepted. So what we did here is we wanted to understand how certain neutralizing antibodies act differently versus some previous antibodies that don't neutralize all variants of COVID. And in collaboration with my colleague Derek Wilson and this pharmaceutical company, Icosagen, what we showed is that with this neutralizing antibody binds to the spike protein, it transmits information down to critical regions, and this leads to the shedding of something called S1 and S2 subunit. And of course, I should say, I didn't get a chance to talk about allostery. There's a huge potential for drug discovery, and it's really underused. The other things, as I said, we're thinking about, we're working on is using these rigidity-based prediction methods for the accuracy of protein structures and how we can design rigidity algorithms and then combine with reinforcement learning, some previous studies, to kind of fix and modify the rigidity of the structure in order to say the structure should look like the one on the right rather than one on the left. And final slide, this is ongoing work, and I cannot show all the details, but there's been, so if you run AlphaFold and you have a structured protein, it will give you this kind of high confidence score, and it's quite accurate compared to, say, crystallography in cryo-EM. It's trained on that data. However, if you look at these disordered proteins, which make up actually 1/3 of whole proteome, either they're completely disordered or they have highly disordered regions, AlphaFold always gives you these kind of low confidence scores, and it just outputs a few models. However, what's remarkable is that not the predictions themselves, but the distances that is used in AlphaFold, something called histogram, the distances between pairs of residues, even for disordered proteins, matches experimental NMR and SAX data. And this was published just recently by Vendor-Scholar group. So we're utilizing this kind of information to constrain the dynamics, constrain the motions of disordered proteins using these fast rigidity-based samplers of dynamics. And then what we want to do is we want to shift kind of this conformational ensemble to the one that matching better these distances that should be matching the experimental data. So you start with some kind of a wrong AlphaFold prediction, and now using this kind of different reweighting approaches using Bayesian maximum entropy, rigidity theory, reinforcement learning, and other techniques we're exploring, we can get, we believe we can get better sense of these AlphaFold predictions. So I think in conclusions, rigidity theory provides really novel theoretical and mechanistics view of allosteric transmissions. Algorithms are extremely fast and allow us to make major breakthroughs in the future using machine learning and AI. And allostery it is also important, of course, in pharmacology and drug discovery. And I want to acknowledge many experimental collaborators. Scott Prosser, who's here, rigidity theory, computation, machine learning people, and thank you very much.
I have a question. So how the Allostery predictions are affected by the model quality? So can it be used to probe, say, model AlphaFold prediction quality, or just because there is obviously this NMR kind of crystal structures, they are obviously considered to be maybe sometimes better than AlphaFold, but that those resolutions vary, so maybe, I don't know.
Yeah, so if you have a really bad prediction, bad model, then these methods can be sensitive to that. On the flip side, there's an advantage to that. If you have a high model, high prediction model, we can pick up like local interactions, perhaps even part of a drug or ring, say this part is important for ulcer, this isn't. Meanwhile, some other models, when you just coarse grain too much, you don't get those details. So you can still get good predictions even for bad models, but the quality of prediction goes up tremendously when you have certainty in the model.
Right. Yeah, I'm thinking about those loops, because loops are, like as you were showing, they are kind of quite important sometimes, but sometimes they are just not resolved in those crystal structures.
Right.
But yeah, with AlphaFold, it's kind of...
Yeah, so AlphaFold, you always get low confidence models, yes.
Yeah, I have a question. Let's imagine in a protein that you have a deep and extended hydrophobic pocket, so that interactions are dictated not by hydrogen bonds, but rather by some kind of, large surface area contact. You could also think about, residues that sort of switch between different charge states, things like that. How do you incorporate those sorts of things into your topological map?
Yeah, so I didn't talk about the details, but in addition to hydrogen bonds, we do have like hydrophobic estimates, hydrophobic contacts predictions as well. So for example, if you have carbon-carbon or carbon-sulfur nearby, you could put some extra constraints there. Yeah, so there is ways to tweak this. And I should say, the constraints that we have here, or the model, is really an all-atom atomistic, including hydrogens. But yes, we're kind of, the quality of the input is very important.
One question from Zoom. Maybe it's not so related, but what features do you anticipate will be implemented in AlphaFold 3's next version? So what features do you anticipate will be implemented in
I see.
Alpha 4.3's next version?
Well, as I said, I think the next thing is to get better predictions for small proteins that were not trained on, say, NMR structures. The other feature would be improvement of quality. The other would be to incorporate dynamics. I think dynamics would be next, training on. But it's hard to get a lot of dynamics data because MD simulation is slow. So definitely improvement of quality and incorporation of flexibility and dynamics.
So what specific AI technologies would be required for this?
Well, I think training large language models and understanding this.
Okay, thank you very much.
Like a question about kind of related to this AlphaFold, because there was AlphaFold 2, but now there is AlphaFold 3, and then there was papers that about using the answer method that, well, it's kind of a good, and then it's kind of practically kind of it is very accurate. But how about AlphaFold 3 predictions? How good are they? Are they better compared to AlphaFold 2? Or
I mean,
rigidity.
I think there was a paper where we showed that AlphaFold, was it 2 or 1 or 2? That the AlphaFold 2 predictions on NMR structures are actually better than most NMR structures. And the reason is that the AlphaFold structures were trained on crystal structures, and crystal structures tend to match the NMR data. When I mean NMR data, I don't mean the NOE constraints. I'm talking about the raw input data, which we use for answer, which is the chemical shifts. So we didn't compare it against AlphaFold 3, so I don't know.
Okay, so thank you very much. So now we are in coffee break.
Thank you for your coming. And first of all, I would like to express sincere thanks to Professor Tsuda and everybody who are related to today's event. And I'm Masato Sumita, a RIKEN researcher at RIP. And today I will give a talk about generative AI and inverse problems in chemistry. I'm not sure what kind of people will listen to this talk, so then I will not go into detail, generative AI and chemistry, and just only the focus on the relationship between them and via inverse problems. It's okay? And I would like to start from this picture. My friend ordered just to make only a pictures, right? So then I made this slide, and I draw this picture. And this is a butterfly, this is a cat. Probably somebody may say that is not the cat, that is not the butterfly, because this is a draw by my imagination. So more specifically, this is a cat, and this is a butterfly picture inspired by butterfly and cat. Understand? So then I would like to explain how to draw, not to use the tools, just only the processes. Okay. So when I was a child, there are a lot of cats at my parents' home. So then my friend, not sorry, my parents teach me this is a cat. Okay. So then I can realize when I look at a cat, I can recognize a cat like this. So then I can image a cat and draw image as a cat. Okay. So probably you can understand we can draw something we know basically. Okay. But yeah, there's two problems. One is forward, just on the target observations. Probably if you are human, there is no problem because we have that function inherently. Okay. But on the other hand, the inverse problem, target generation, not. Probably everybody can understand what I said. So then I would like to explain how to solve the inverse problem. Surely under several limitations, my skill and time, money, and etc., And under the several limitations, I image the cat's pictures. And then I try to draw something, draw my imagination. And the first trial, I evaluated that first trials. This picture is nice or etc., etc. And then based on these evaluations, I again, that's not. We consider some kind of poor parts that should be fixed. And then sometimes rewrite. And then again draw and evaluate. Probably I repeat this cycle, and then this picture produced. If you are gifted person, there is no need to repeat this cycle. But I'm not, I don't have no talent, so then it's necessary to repeat that cycle. So many fails, okay? And probably you can understand to solve the inverse problem, human must make an effort. Probably you can understand. So recently, generative AI appeared, alternative to creativity. But there's no difference with a human. First, this AI should be know the cat. So then we train this AI using this picture of cat. So then if we order draw a picture of a cat, so then this AI can draw a picture like this. So generative AI can draw something previously learned. Yeah, there's no difference with a human. So, but probably you may realize that we don't need to do any efforts anymore. But yeah, so this appearance of the AI might be we can become lazy. We don't need to make any efforts. But unfortunately, not. My talk is about that. And now we are arrived at the chemistry. And chemists are also dreaming. Generative AI that could generate molecules having something demanded like this. If the chemists ordered a molecule of something, so then this generative AI generate molecules like this. This is a dream. But unfortunately, in the real chemistry like this, I hope there's no human who can see molecules. so then it is impossible to see the molecules. So it means there's nothing to run. Okay, so then it is impossible to draw molecules. Probably you can understand. And so it is simply impossible to even see the molecules like this. So then generative AI not so effective in the chemistry. But chemistry is one of the discipline that suffer us. And so then chemists developed many instruments to see molecules. In the beginning of chemistry, the combustion analysis is a major. This analysis clarifies the compositions, just only compositions. And next is a mass spectroscopy. Yeah, this spectrometry, sorry. Mass spectrometry clarify mass of molecules. Sometimes used to clarify whole molecular structures. And infrared Raman spectroscopy is very important spectroscopy. But this spectroscopy just only clarifies a fraction of molecules, functional groups. That's the characteristic moiety of molecules. And nuclear magnetic resonance NMR spectroscopy also clarify very useful to clarify the whole molecular structures. And X-ray and several particle scattering also clarify whole molecular structures. But it is necessary to crystallizations. And as already probably know the previous presentations, as molecular size growing, it is very difficult to identify molecular structures. And here is a NMR spectra as an example. And this spectrum indicates these molecules. Yeah, probably non-specialists, it is very difficult to this spectra to decode these molecules. So spectra need to be decoded by specialists. Okay. So visualizing molecule is a typical inverse problem called identifications. Okay, so chemists should be learn the formulated knowledge. Yeah, that is written in the chemistry textbooks. And then getting a spectra, major spectra. So these chemists can decode this spectra to the molecules like this. So chemists identify molecule using their expertise. Okay. So let's see the inside chemist's brain. And spectra is some kind of histogram molecular parts. Surely depend on the spectrum species, but for the NMR or IR like this. And this and formulated knowledge tell us the where the these parts appear on the spectroscopy. Okay, so then we can based on this formulated knowledge build molecules, and then evaluate the built molecules on the subject of consistency with spectrums. Okay, and based on this evaluation, they are again considered. Yeah, surely one cycle is enough, but sometimes multi-cycle needed. So, but anyway, you already realize that the molecular identification is some kind of good target for generative AI as a molecular identifier. So if we can make an AI that is trained by spectra of molecules, yeah. So then this AI get some molecular spectrum as an input. So then it is easy produce how to say decoded to the molecule like this. So then generative might be a good alternative to the chemists. There's no need to the repeated cycles. Okay. So then we are also trying to make some AI as an identifier using IR Raman spectroscopy. This is a collaboration with Futomitsu and IR Raman spectrum already said used to identify functional group, not the whole molecular structures. Sometimes used, but so then I try to use IR Raman spectroscopy to identify whole molecular structures. And IR Raman indicates the molecular vibration. So then spectra is very complex. Yeah, like this, many zigzag. So then vibration spectra typically, yeah, it is very difficult too complex for human to decode. Okay. So generative AI for IR Raman spectra is like this. Yeah, this is some kind of details. And yeah, training data set is almost 500,000 molecules. And please take care for that up to the 9 heavy atoms, CNOF. and IR spectrum obtained from the density function theory. And algorithm encoder-decoder systems, both sometimes use transformer, sometimes we use multi-layered perceptrons. And IR Raman spectra have a complementary relationship. I will not go the detail about this. Why there is a two-type vibrations spectrums. Yeah, but there is two-type vibration spectrums. But anyway, if we use the both spectrum, it might be accuracy increased. We expected that. So this is our result. And sorry, yeah, this is a result. The spectrum of molecular consistence composites of fewer than 9 heavy atoms. Yeah, this is the input. And if we use only IR, almost 50%. And if we use a Raman spectroscopy, 63%. But if we use both IR Raman spectra, almost 80%. So then IR and Raman shows a superior result than single IR Raman spectrum. So AI can reveal a whole molecular structure from IR and Raman spectra. Okay. So we are trying to investigate the extrapolation ability of the AI. So, and next is the input is a spectra of a molecule consistent of more than 10 heavy atoms. Training data set is consisted of the 9 heavy atoms. So then this spectral input is some kind beyond the training data. So when the number of heavy atom is 10, that case 13%, and the number of heavy atoms 11, like this, 0%. So then we can conclude that generative cannot generate a molecule beyond the training data. Yes, this is a good result, I think. So for the IR and Raman spectra is very easy to define the beyond, because the number of vibration peak is dependent on the number of electrons. So then yeah, it is a very easy case, but typical molecular structure is very difficult to what is beyond. And so then it's necessary to the whole molecules. So how many molecules in chemical space? Chemical space means the molecules are housing. And known molecules 10 to the 8 powers. And yeah, according to the PubChem. PubChem includes all molecules known, yeah, synthesized, successfully synthesized molecule, but sometimes strange molecules included. And so on the other hand, the expected molecules are like 10 to the 60 powers. So yeah, surely this number is limited in the drug molecules. If we remove this limit, probably countless, yes. So most of chemical space is unknown. Surely it might be 10 to the 8 molecules might be enough to whole chemical space, but there is no guarantees. Okay, so this is my conclusion. AI and machine learning is not used for chemical research. Okay, yeah, and yeah, but research should be done for the future. So then for the chemistry should be developed as something new, something novel. So then AI is not for the, basically not for the chemistry research, okay? So I always say so that I will kicked out from the AIP center. so then yeah, hereafter I will show some of the examples of how to use machine learning and chemistry, okay? first example is to generate negative data. Not we are aiming to generate something superior something. Just only that generate the negative data. And chemists don't bother to measure low performance matters. You understand? Because they are always interested in the high performance something. Yeah, otherwise it is impossible to get the high impact journal. Okay, so then probably they take some measurement results, but they are not published probably. So I think data are always biased, and yeah, prevent getting the unbiased comprehensions. Okay, so we are trying to use generative AI to mitigate data bias. Okay. Yeah, this is a typical example, I think. This is data for the gas separation polymers. Yeah, this is a Robinson plot. I'm not sure how to pronunciations. And but anyway, I'm not going oh, sorry. I will not go to the detail of this plot. Yes, but yeah, I just only show this data is biased. That is the data is localized around the red lines. Okay. So then a conventional suggestion is the importance of rigidity of polymer. Sorry. So based on the experimental chemists, they always suggested rigidity is very important based on this biased data. So we are trying to use generative AI to generate sequential data of gas separation polymers like this. We use ChemTS V2 as a generative AI. I already explained this generative AI elsewhere. I'm so fed up with to explain this generative AI. If you're interested, please read this paper. And this ChemTS is this generative AI easily switch the evaluation systems. So here we use a prediction models. That is a machine learning prediction models. And training data set is like this. I will never go details. And this is a generated results. Generated polymers data is described, written in the orange plots. And we are going to discuss the structure and gas separation. Yeah, now ongoing. And now I'm writing a paper. We are writing a paper. I'm just pushing first author. And so then chemists always are interested in the structure and property relationship. Because chemists always consider the material design based on the molecular structures. So then they are always try to get the structure and the property relations. So I think it is possible to generative applicability depend on this relationship, that is structure property relationships. If the structure formulated well known and the mechanism also known like this, yeah, AI is okay. Please go on. And yeah, as already shown, the example is a spectrum system. Yeah, it might be good to target. But for the mechanism unknown and structure formulated cases, I can't recommend to use AI because in this case, probably structure is decided by the chemist's intuition. So then if we apply the AI, we are trapped by the local minimum. So then I can't recommend. And if the mechanism unknown, and the structure unformulated. That is already shown, the gas separation systems. So this system is also, I can't recommend to use a gas AI. But yeah, I already showed that how to use AI. First, we use the gas separation polymer, move to this region, okay? So then, yeah, it is very difficult. And finally, mechanism known and the structure of formulated cases like this. Yeah, also, I can't recommend to use AI. So generative AI is not applicable to the unformulated, unknown systems. Yeah. So then everything, almost everything, we should not use AI for the research. Okay. But finally, I will show example structure unformulated, but mechanism known cases. That is a light emissions. And this light emission is structure unformulated, but mechanism known probably. Yeah, this is a very, very simple mechanism. For the practical light emission molecule is very complex, but yeah, more complex. But this is a very, very simple. But computational chemistry can formulate the mechanism, but human cannot formulate the molecular structures. So then practical molecule, practical light emission molecule is used. That is already light emission. Okay, so then this is a design light emission molecules details. I already said ChemTSV2 we used, and for the aberration we use automated density function theory. Okay, so this is a result. Totally 8 molecules we measured, experimentally measured. This is a known molecule, seven molecules known, and this is a unknown molecule. Unfortunately, AB molecule is not for light emission, but totally 75% generated molecules show light emission. So simulation plus AI can design unformulated materials from scratch. Yeah. Like, okay, final, my conclusion like this. AI and machine learning are based on past data, so novelty is given by humans, okay? Like imagination is very important, like this cat. And we should not be lazy, okay? We should always consider when you use AI, okay? Thank you for your attention.
Okay, I'll give you a problem from my lab. Let's imagine you're doing drug discovery on a target. But in this case, you have hundreds, if not 1,000 data sets of molecules that you know bind in some way to the target and give some kind of output. But you want AI to take over at that point, and you don't want to tell, in this exercise, you don't want to tell the AI anything about the target, just the input and the output.
Yes.
I mean, do you just step into a black hole if you try to do something like that?
Yeah, that's right. But if you have some kind of simulations help, it might be overcome. I'm not sure what's happened, but yeah, it might, surely that depends on the simulation's accuracy. For example, drug discovery cases, already many docking simulation is used, but their accuracy is very low. Probably 10% active some kind of small molecules are obtained. So then surely depend on the simulations. And expand the training data is, how to say, depend on the simulation's ability. Yeah. Okay.
See, it's a common problem in fragment-based drug discovery because maybe you have a lot of targets, you're doing high throughput, and you have a big library. And so you can't rely on docking with your library because you don't know if you're docking an allosteric pocket or anything. You actually don't want to even, try that piece. What you want to do is throw many, many, many thousands of fragments at the target, and then you would get input, you would get output, you would get binding data, and maybe even activity data.
I'm not prohibited to use AI. Yeah, but sometimes a human always forget something, and how to say, it is very difficult to everything to see that. So then it is sometimes human drop something important. So that in that case, neural network like AI is very useful to. So then I'm never prohibit. Let's try, but sometimes you get the good result, even if the database biased. Yeah, sometimes you get the good. Yeah. So that it depends on the stochastic. Okay.
I have a question.
Okay.
Thank you for a very interesting presentation.
Yeah.
Presentation.
Please,
Yeah. So I have a question to professors questions as well. So as you mentioned, the search space is quite vast, right? So like 10 power 66, right? So the role of the initialization, right? So and then how we kind of explore this search space, right? How does it like affect? So kind of in ChempTS, it's a Monte Carlo research. With Mitsuishi-san, it's a beam search, but so how does it I think the efficiency of the exploration of the search space also kind of might be important. So what are your feelings about this?
Okay, your question is a little bit over my head. Please talk about that. Okay, but anyway, I would like to push you. Please hurry up the analysis.
Okay, so one thing from Zoom is, could you provide a detailed explanation of how to use ChemTS for generating novel molecular structures?
That's the big problem. Sorry,
you don't have something.
Yes, today I will not prepare some kind of slides. Yeah, but it's something.
We did it in the.
Yeah. But the how to say
in the past, actually in the past we
did, this review paper is open access. So then everybody any can see this paper. Please read this paper. Okay.
Okay. Maybe another question is, ChemTS relies on the reward function to guide molecular generation. How do you balance the trade-off between the computational cost of evaluating molecules, especially when integrating with the computationally expensive methods like quantum chemistry or docking simulations, and the efficiency of the MCTS exploration? Yeah, maybe. Yeah, so this is very. But actually, for us, ChemTS is very fast method. Actually, it doesn't, or mostly it doesn't consume any computation actually, because so actually the most important point is to reduce the number of simulations, because simulations are much slower. No. Yeah. So actually, there's no strategy to take the balance, because for our case, search is negligibly fast. Yeah, but anyway, thank you very much for asking.